to. Here's now the process called translation. We've made this RNA, and now we're going to use it to make proteins. Well, now we have some different kinds of RNA. First of all, the ribosome, this is the ribosome, right? Small part, large part. The ribosome is made of RNA itself. That kind of RNA is called ribosomal. It's also made of some proteins. It's a thing we call a complex. Two or more things stuck together is a complex, right? And so that's one kind of RNA. It's made by transcription in the nucleus. Then we have these things which here are represented by like a barrel with a bend in it. We'll look at them more closely. And you can see they have 3 bases sticking out of one end. Well, they're actually made totally of RNA, so they got a lot more bases squiggled up in this part, right? But that's the kind called transfer RNA because they're going to pick up an amino acid and transfer it to the place where they get connected to each other, which is a ribosome. They're like little delivery trucks. We're going to make another analogy now, okay? If you look at the ribosome in this picture, the ribosome is kind of like a parking garage because that's where these delivery trucks are going to park. They're going to park, deliver their package, amino acid, to a position that is right next to the one that it's going to bond to. Did you follow that? They're acting like an enzyme. The ribosome is a catalyst. This is kind of like an active site, but since it's not all protein, we don't call it an enzyme and we don't call that an active site, but it's the exact same idea. It's the right shape to fit transfer RNAs, and it helps line up 2 things that are going to be put together by, guess what? Dehydration synthesis, right? And these bonds that are forming here are what kind of bonds? Peptide bonds, right? They're between proteins. They're between the carboxyl carbon of one and the amine nitrogen of another. See how much we review as we go. Okay, so the RNA that we're going to focus on the most is the one that's carrying the message, the pink one here. We've already said that that's called messenger RNA. So now we have ribosomal RNA, messenger RNA, and transfer RNA. And in AP biology, you'll learn about a whole bunch of others that do a little more complicated things that we're going to skip here. Okay, so here's 3 models of transfer RNA, right? And here you see that this is RNA, right? This yellow band represents the sugar phosphate part. Each of these little yellow things represent one of the bases. That represents hydrogen bonding. And you see here that sometimes, in fact, it's quite often, that RNA binds with itself to make a double-stranded parts, right? Here's a single-stranded part, and here's a double-stranded part. When it's made, it's all single-stranded, but then it quite often double strands up in places by complementary bases pairing. If you see, all of these are complementary base pairs. So what this molecule looks like is kind of like a cross, right? A clover leaf, a cross. It's got a bend in it, right? Here you see a bend in it. That this very simple model shows the bend in it. But the key parts of this molecule are 2 ends. One end where the amino acid attaches, right? They didn't put one. I'll put one. I'll put proline here, right? And then the other end that's got 3 bases that stick out, right? Remember, there's bases all over this molecule, but these 3 are a key. They are called the anticodon because they are going to stick to a codon. How are they going to stick to it? Because it's complementary. Which codon would this anticodon stick to? The codon that says UUC, right? Remember, codons or anticodons now are made of RNA, so they don't have any T's, so this would not be a T, it would be a U. UUC is the codon on a messenger RNA molecule that this little delivery truck is going to use to park at. So a codon, in our analogy, is kind of like that parking sticker at somebody's parking place that says Mr. Bigsby parks here. Okay, so I'm going to practice that in class with you, right? Which amino acid is attached to the transfer RNA? I'll do it right now. Here's the transfer RNA, right? and it's got some amino acid at one end. That's what the question is. At the other end, it's got like CCU, right? Which amino acid is here? You got to use your chart to figure that up. But you don't look this up in your chart, right? Because your chart is not made to match an anticodon with a particular amino acid. It's made to match a codon. So what do you need to look up in your chart? You need to look up the codon that this is going to attach to, this anticodon. So now it's simple. It just takes two steps. You have to do what a cell has to do. You got to take this message and figure out its complement, and then look up the amino acid in the book. I forget what GGA is. It might be glycine. I forget. Okay, so here's a little bit more detail. We call these three parking spots in a ribosome the EA and P spots. The A and P are the ones we'll focus on a little bit, but this is way past EOC detail stuff, but not for this class. Not. So we're going to watch this in video. I don't even remember if I have a diagram coming up here, but we have a process that's very similar to transcription. We are going to start in one place. We are going to start making a polymer, and we're going to make it longer and longer and longer, and then we're going to stop in another place, and we're going to call these parts of this process by the same words as we called it before. I've got some good animations that show this, but here it showed in one kind of way the starting, the initiation, the place where it starts, okay? This again we said is always on a start codon, and that's always AUG, okay? But notice it's not at the end of this messenger RNA. This messenger RNA has other bases here, right? But when an AUG is reached, that's where the ribosome attaches. The ribosome is now fully attached, small subunit first, large subunit next, and it attaches so that that site, that little place where these can park, the site called the P site, can accept a delivery truck transfer RNA that attaches to the AUG codon because its anticodon is UAC, and it always carries methionine. And that's what we get started. This takes some energy. We'll not worry about this. This is a little cousin battery of ATP. It just has happened to be the one that evolved to power this process, right? And now we're ready to go. And now we have an open parking spot right next to this. And so now another transfer RNA is just going to come in here, park, deliver its amino acid right here. They're going to bond, right? And then this whole thing is going to shift. So now we're here, right? We fast forward in time a little bit. A transfer RNA that delivered its amino acid and had it attached to another one, which before had been attached to another one, is now holding that chain. Now we have, I skipped the middle parts. I'm going to have to do this in class because I kind of did it the last time. But we've reached, we reached the stop codon here. And you notice that a stop codon, a protein release factor attaches here, it parks here, but it doesn't carry another amino acid. It's not a transfer RNA. When it attaches and it tugs and pulls, all of this stuff comes apart. This last transfer RNA that was holding the entire protein, it comes out of the P site. The two subunit ribosomes come apart. The polypeptide is now done. It might get processed later, like in the Golgi apparatus, right? In fact, it almost certainly will. So we'll attach that part there. And there we have the termination stage. We kind of skipped over I thought there was a slide showing the elongation stage, but I kind of described it. It's just one after another after another. These delivery trucks come and they fill a spot, and then we slide down. We'll see that in video in class. Okay, so There's the one we're going to look at. We're going to practice diagramming these in